Muy buenos días a todos. En nombre y representación de la Federación Centroamericana y del Caribe de Laboratorios Farmacéuticos de Investigación y Desarrollo, FEDEFARMA, les damos a todos ustedes una muy cordial bienvenida a esta sesión virtual donde abordaremos una descripción general de los productos de terapias avanzadas. En esta actividad, organizada por el Comité de Asuntos Regulatorios y Productos Bioterapéuticos de FEDEFARMA, es parte de nuestro compromiso de promover e impulsar el acceso a los avances de la ciencia médica que permitan a los pacientes tener una mejor calidad de vida. Mi nombre es Douglas Rodríguez Calderón, miembro del Comité de Asuntos Regulatorios y Productos Bioterapéuticos de FEDEFARMA y gerente de Asuntos Regulatorios Estratégicos y Externos en Janssen para Venezuela, Centroamérica y Caribe. Será un gran placer para mí y un honor actuar como moderador en esta actividad. A continuación, tengo el honor de presentar a Paula Vargas San Martín, directora y cabeza del Departamento de Asuntos Regulatorios para Pfizer en Centroamérica y Caribe y presidente del Comité de Asuntos Regulatorios y Productos Bioterapéuticos de FEDEFARMA para el periodo del 2021, quien brindará seguidamente las palabras de apertura a esta sesión. Paula, adelante.
Hola Douglas, muchas gracias por la introducción y buenos días a todos. Para nosotros es un privilegio contar con su presencia el día de hoy en este webinar. Como muchos de ustedes saben, FEDEFARMA es la Federación Centroamericana y del Caribe de Laboratorios Farmacéuticos. Es una entidad gremial que hoy está compuesta por 19 compañías de investigación y desarrollo. Compañías que suministran medicamentos y terapias innovadoras a lo largo de toda nuestra región de América Central y Caribe. FEDEFARMA desde los años 70 viene apoyando de manera activa las autoridades y gobiernos a través de una cooperación técnica para el bienestar de la población. Una parte muy importante de nuestra misión, como podían ver en el video introductorio, es trabajar de manera conjunta con gobiernos, autoridades de salud, entes regulatorios, comunidad médica, científica, academia y por supuesto nuestros pacientes, para facilitar el acceso de esta innovación y garantizar así la calidad, la eficacia y la seguridad de los medicamentos que son consumidos por nuestros pacientes, siendo así un ente de referencia y principalmente apoyo técnico a nuestras autoridades de la región. Esta mañana estamos reunidos en este webinar, creo yo, para ver un tema fascinante para muchos de nosotros, que es una descripción general de las terapias avanzadas. Nuestro gran objetivo es poder compartir todo este conocimiento con todos ustedes. Y dentro de los tres principales temas que vamos a intentar abordar en esta mañana es pues hacer una mirada a esta introducción de terapias avanzadas. Es realmente importante que conozcamos los conceptos básicos de estas terapias y podamos entender cuáles son esos productos que hoy forman parte de esta categoría. Dentro de los que podemos mencionar terapia génica, terapia celular, ingeniería tisular y terapia avanzada combinada. Otro de nuestros grandes objetivos esta mañana es poder determinar cuál es esa diferencia entre los productos de terapia avanzada y los que conocemos hoy como medicina tradicional, así como sus beneficios terapéuticos. Creemos como chapter que si no entendemos estas definiciones básicas, no podemos pretender asegurar la construcción de un marco regulatorio sólido y seguro para el ingreso de estos productos cuando estén en proceso de registro en nuestros mercados. Y finalmente, uno de los temas que creo que como audiencia, en muchos casos del área regulatoria va a ser de gran interés, es entender cuál es ese marco regulatorio que hoy ya existe en Europa, en Estados Unidos e incluso en países de América Latina como Brasil y Argentina. El poder hacer una revisión de estos marcos regulatorios nos puede dar una visión clara de hacia dónde debemos dirigirnos en un momento en el que necesitemos empezar a generar estos marcos regulatorios para productos que parecían que estaban lejos de llegar a nuestra realidad, pero la realidad de hoy es que ya están en medio de nosotros y necesitamos asegurar que se registren de manera apropiada. Este acercamiento con la región se realiza con el fin de profundizar en el rol fundamental de la innovación farmacéutica y médica en los sistemas de salud e identificar algo muy importante para todos nosotros, que son aquellas oportunidades para garantizar el acceso oportuno de los medicamentos y de las terapias innovadoras. Agradezco grandemente a cada uno de los expositores que nos acompañan hoy en esta mañana por haber sacado ese espacio en la agenda, por poder compartir todo ese conocimiento con cada uno de nosotros y así mismo a la audiencia por confiar una vez más en un webinar de FEDEFARMA y acompañarnos esta mañana en este tema. Muchas gracias. Douglas, te regreso la palabra.
Muchas gracias. Paula. Nuevamente agradecemos a la presidenta del Comité de Asuntos Regulatorios y Productos Bioterapéuticos de FEDEFARMA por la apertura. A continuación, vamos a brindar unas breves instrucciones para habilitar la interpretación simultánea al idioma español y realizar las preguntas o comentarios en la plataforma. Referente a la interpretación simultánea, la mayor parte de la exposición será en idioma inglés. En el caso de que desee escuchar su exposición en idioma inglés, deberá seleccionar el icono de la interpretación con la figura de un mundo, ubicado en la esquina inferior derecha del panel. Posteriormente, seleccionar el idioma inglés y hacer clic en la opción que indica silenciar el audio original, con lo cual la interpretación quedará habilitada de manera inmediata. Recuerde que en cualquier momento puede alternar o escuchar las ponencias en inglés o en español, por cuanto la interpretación simultánea estará habilitada durante todo el webinar y en todo momento. En cuanto a las preguntas, estas pueden ser enviadas en idioma español a través de la casilla habilitada para preguntas y respuestas, que podrán encontrar en la parte inferior de su pantalla. Las mismas serán atendidas por los expositores al finalizar todas las ponencias. Dadas estas breves instrucciones, a continuación pues, tengo el honor y el agrado de introducir a todos los expositores que nos compartirán tan relevante información esta mañana. En primer lugar tenemos a Susan Stevenson, ella es directora ejecutiva para la área terapéutica de hematología de Novartis. Tenemos luego a Orin Temkin, quien es el líder global de área terapéutica oftalmológica a nivel de asuntos regulatorios también de Novartis. Tenemos a Miriam Fuchs, quien es líder global de área terapéutica oncológica, terapias génicas y celulares de Novartis. También tenemos a Lauren Stark, experto de inteligencia regulatoria para cambios de manufactura y controles de Novartis. Tenemos a Chin Koener, director ejecutivo de asuntos regulatorios para desarrollo de políticas regulatorias. Mireille Müller, director de políticas e inteligencia regulatoria, asuntos regulatorios para desarrollo global de políticas y medicamentos. Y finalmente, Olivia López, quien es líder de asuntos regulatorios para estrategias de nuevos productos en Latinoamérica y Canadá en Novartis. Sin más preámbulo, cedo la palabra a nuestros expositores para que inicien con sus ponencias. Susan, la audiencia es tuya.
Hello, can you hear me now? Can you hear me? Okay. Someone verify. Can anybody hear me? Can you hear me now? Can you hear me now? Apologies. Should I just keep talking?
Yes, we can hear you.
Oh, okay, good. Thank you so much. Okay, go to the first slide, please. Slide 3 of the presentation. Are the slides being presented? I don't see them.
See the presentation.
I don't see the presentation. Oh, okay, here we go. Okay, great. Thank you. Thank you so much. Thank you for the introduction. Today, I want to provide an overview, a short introduction to advanced therapies, review the progress that's been made over the last 3 decades of research in developing these next generation medicines, and give you a brief introduction as to their various tools and how they're being applied in the field. Next slide, please. oh, okay, slide, keep going. Next slide. Great. So advanced therapies are the future for patient care and represent the next generation of innovative medicines. Advanced therapies refer to new medical products that use gene therapy, cell therapy, and tissue or cell engineering, and they can be used to treat diseases or injuries and have huge potentials for the future of medicine. Now, recent advancement in technologies for the development of these biological therapies have resulted in a gradual shift towards personalized medicine from the conventional one-size-fits-all approach as seen for small molecules or antibody therapeutics on the left-hand side, to a more complex drug product that falls into this advanced therapy category on the right-hand slide, part of the slide. The advanced therapy medicinal product landscape is one of the most active areas of drug discovery research. These products offer solutions for conditions with little to no therapeutic alternatives available and is one of the major growth drivers for the market. Advanced therapies are currently really in its nascent stage. It's a constantly developing area. And as shown on the next slide, the number of investigational new drug applications or INDs in the United States for these therapies continues to increase. Next slide. This is shown here with an analysis of the clinical trial pipeline in a press release by the FDA last year, showing that in 2020, there were over 900 investigational new drug applications or INDs from ongoing clinical studies and a variety of serious diseases for cell and gene therapy. These are being developed in different areas like blood disorders with lentiviral gene therapy or CRISPR gene editing of stem cells for sickle cell disease and HIV, a variety of neurological disorders with local delivery into the central nervous system or local delivery directly into the eye for inherited retinopathies, as well as different indications in the musculoskeletal disease setting. Now, the majority of these clinical trials are being developed in the oncology setting, with over 64% or so of those targeting in oncology, with a variety of those targeting blood cancers. The results, now given this breadth of activity, it's predicted to result in about 10 to 20 gene therapy medicines for approval by the year 2025. This is a very exciting time in the cell and gene therapy space. Now, this productivity, if you will, is a result of decades of research into technologies needed for cell and gene therapy. The evolution and productivity from these many years of efforts is shown on the next slide. Next slide, please. Now, the cell and gene therapy field started in the early 1990s with the first ex vivo hematopoietic stem cell therapy using one of the very first vector systems available, which was a retrovirus. Now, this is the Gartner hype technology curve, and I like to show the evolution of the field using this because this represents what happens in the discovery and adaptation of a new technology. Now, at the beginning, there's a lot of interest, there's a lot of unknowns, and it's used very widely, and this results in an increase of activity using the technology. And then as we learn about what gene therapy technology could or could not do, we realize that there's a lot more work to do to understand and improve what was learned from these early clinical experiences. And this is shown by the big drop of interest in the use of the technology. This trough is called the trough of disillusionment in this hype curve. Now, this is where the gene therapy field took a big step back in the late 90s and realized it had to further understand and develop and refine the delivery platforms of the technologies that are being used to deliver the genes and modify the cells. And as a result of learning about not only how to use it to get robust efficacy, but most importantly, learning about its safety profile, we then started applying the technology in situations where it had its best chance of success. And this is shown by the upslope now of approved medicines starting in 2016 with Strimvelis, followed by two chimeric antigen receptor T cell therapies in 2017, the first AAV gene replacement therapy approved in the United States in 2017, and then the most recent approval being an AAV gene replacement therapy for spinal spinal muscular atrophy in 2019. And now this is where we're in this upslope. This is called the slope of enlightenment, which means we have a greater understanding of the technology and its risks and benefits. And now we're hopefully entering this plateau of productivity where we will continue to see a steady string of new approvals for these advanced therapy medicines. And hopefully we'll be able to see these anticipated 10 to 20 new approvals by the year 2025. Next slide, please. So what are advanced therapies? So as listed here on the left, advanced therapies are known as cell and gene therapies, regenerative therapies, or advanced therapy medicinal products. They can contain or consist of engineered autologous or allogeneic cells or viral vectors that can recode cells to fight against diseases or correct genetic defects. Advanced therapies are more complex than the traditional biologic therapy, like a monoclonal antibody. They're more difficult to manufacture, and as with any medicine, they must be manufactured under GMP conditions. The different products are listed here on the right-hand part of the slide and are characterized as being either an ex vivo modified cell therapy or a viral vector for a gene therapy, or even can be combined with a novel device for delivery. The next slide shows you the delivery of these advanced therapies shown schematically here. And again, cell therapy generally means that cells are removed from the patient, modified in some way, and then delivered back into the patient. The cells can come from the patient, and this is referred to as an autologous cell therapy, or the cells can be taken from one person, expanded, modified in some way, and then used to treat many patients. And this is referred to as an allogeneic cell therapy. On the right-hand part of the slide, in general, a gene therapy refers to a delivery of a gene directly into the patient and can use a variety of different delivery platforms of either using a viral vector, an engineered viral vector, or a non-viral based delivery system. Slide 9, the next slide, gives a bit more detail for each of these approaches that are listed here as either ex vivo on the left part of the slide or in vivo on the right. Now, this diagram is a very simplistic representation of what's done. You'll see in the next presentation the complexity of what's actually involved in developing and implementing an ex vivo CAR T cell therapy. Now, over the decades of research in the cell and gene therapy field, the optimization and innovation on each of the delivery platforms have significantly advanced. And in each area, whether it's ex vivo or in vivo, a dominant delivery platform has emerged. Now, when treating proliferating cells ex vivo, the cells must be permanently modified, since these cells are proliferating and growing. So most approaches either use an integrating vector system like Lenti or use different genome editing tools like CRISPR to introduce a permanent change within the genomic DNA of these isolated cells, such that when the cells are infused into a patient, that genetic change is permanent. Now, on the right-hand part of the slide, there have been many applications of in vivo gene therapy over the years, and the use of the adeno-associated vector system has become the most used vector delivery platform for gene replacement therapies. Depending on the route of delivery, local versus systemic, and the type and the turnover rate of the target cells, the cargo that's delivered can be varied, including replacing the gene that is defective with AAV, then this will provide for long-term expression. AAV can also be used to deliver different types of therapeutic constructs, including gene editing cargoes to correct or repair the defective gene, as well as delivery of other therapeutic proteins that can use to be modified the phenotype. The next slides show these vector delivery systems in just a bit more detail. Now, each viral vector delivery platform are used for distinct clinical applications. And as I mentioned before, lentivectors will integrate into the genome when introduced into cells ex vivo or in a dish, and that this will result in a permanent modification. Now, as a result, they've been used exclusively in ex vivo stem cell therapies for genetic diseases or ex vivo T cell therapies. Now in the middle, AAV vectors do not integrate, so they've been used to deliver genes into non-proliferating cells or terminally differentiated cells like neurons or photoreceptors, since the DNA of these vectors will persist and continue to express a functional copy of the gene that's being replaced. Now, as shown here, they're used primarily in genetic diseases. Last, on the right, is the adenovirus delivery platform, and this system also does not integrate, so provides for transient expression and exposure. Now, this was one of the first viral vectors used in the gene therapy field, but now is used primarily in vaccine antigen expression, including recently even for COVID-19. Its other major use is oncolytic vectors for oncology. Now, this slide represents decades of research and progress in developing these viral delivery platforms and our understanding on how best to use them to develop robust therapies. Next slide. This last slide to finish just summarizes why we develop these advanced therapies. Now, again, compared to conventional therapies, they're more complex, but this approach with developing advanced therapies provides for the potential for a one-time curative therapy for patients. And it's this transformative potential that makes this of significant interest and a strategic growth area for us. So thanks for your attention. And now I'm going to turn it over to Oren Temkin, who will tell you about how these therapies are different, and he'll highlight some of the complexities of developing these advanced therapies. So Oren, I'll turn it over to you. So thank you.
Thank you, Sue. Buenos dias, good morning. Thank you for the opportunity to speak here today. I would like to continue with Sue's introduction to advanced therapies by talking a little bit about what makes these therapies different from the conventional therapies most of us are used to developing. They do pose some very special development challenges which make us need to carefully rethink the way we develop drugs. Could you please progress to the next slide? Thank you. So this repeats a slide that Sue showed, and I think it's worthwhile emphasizing the difference between conventional and advanced therapies. Our conventional therapies are molecular compounds, typically either small molecules or biologicals, and these tend to be chronic therapies that address long-term treatment of symptoms. When we think about these advanced therapies, gene and cell therapies, these have the potential to address the root cause of the disease. And this is a very fundamental shift from those therapies. So this involves delivery of a new functioning gene where a gene may be missing or not working properly, or the use of the patient's own cells, which can be removed from the body and then reprogrammed and returned, what we can refer to as a living drug. With this ability to address the root cause of disease, we have the potential for a one-time treatment that is transformative and potentially curative. And this is in contrast to that long-term chronic therapy and symptom treatment that we're more used to with the conventional therapies. So these advanced therapies really do hold great promise. Next slide, please. So let's discuss some of these unique features, starting with clinical development. Before we get into the clinic, we have to think about preclinical development, and for many of these diseases, there is a lack of relevant animal models. So we don't always have the same body of preclinical data available as we think about our clinical studies. These therapies will have a condensed or shortened development paradigm where those traditional phases of drug development would likely be merged together into a first-in-human phase 1-2 trial with a limited dose exploration in part 1, followed by a confirmation of safety and efficacy in a pivotal study, part 2, and then long-term follow-up being looked at as a part 3. separately. There are some challenges related to trial execution. A big one is enrollment, especially since many of these therapies focus on a rare or even ultra-rare orphan disease populations where there are very few patients. The merging of the earlier phase and the pivotal studies leads to a seamless trial design where these may be part of a single trial. When we're implementing the trial, at the trial sites, additional training is needed in handling of the gene or cell products, compounding, and administration. These are not your routine tablets and capsules. These require administrations, sometimes through a surgical technique by a skilled surgeon. So the handling and administration of these is something that has to be considered on its own. For the autologous products, individual manufacture for each patient is a concern because we're using the patient's own material, their own plasma. We have to think about an accurate diagnosis by validated gene testing to make sure that the patient actually has the condition for which the treatment is targeted. So that introduces the need for a validated test. There's a critical role for appropriate biomarkers as surrogate endpoints, particularly in neurodegenerative diseases when the disease progression is slow. So this is different from your more routine endpoints. With these products, there's an important role for natural history data to look at the natural course and progression of the disease. And this may be needed in a clinical trial where a placebo or a comparator is not feasible or practical. Lastly, I mention here the need for GMO requirements, which may be more extensive and burdensome for a gene or cell therapy than it is for a more routine product. Next slide, please. This shows some chemistry, manufacturing, and controls, or CMC considerations. These are absolutely critical during the development of a gene or cell therapy. The large majority of these therapies are initially developed in a small scale in academic settings using labor-intensive multi-step processes, requiring iterative development and optimization, scaling up to commercial scale under GMPs in a relatively compressed period of time. So that makes that development of the process quite challenging. The material, the patient material, as used for autologous products, has to be considered, and lots variability of materials that are human and animal derived have to be taken into account, as well as the fact that there will be limited product knowledge in the earlier phases of clinical development. And this is very important to keep in mind as we try to develop a robust commercial product for these advanced therapies. Understanding the product's critical quality attributes and critical process parameters, and the establishment of clinically relevant product specifications has to be done very thoughtfully and also on a shortened time frame. So we have a very good understanding of what the most critical aspects of that process are. We need to develop robust analytical tools for dose and strength, and make sure that we have a control strategy that ensures consistent product quality. When we think about process validation, when we take into account that, again, we could be using the patient's own plasma or material, we may need to include a surrogate starting material from healthy donors in combination with patient material, since those patient materials may be scarce. And lastly, comparability is a key element because there is a high rate of process change and improvement during this period. So those criteria have to be set for how we establish that the old process is equivalent to the new process. And this may happen several times during development as well as post-approval as that manufacturing process naturally evolves. Next slide, please. Over the next few slides, I would like to highlight a few of the transformative cell and gene therapies that are in Novartis's portfolio that we have been developing and registering all over the world. The first of these is onasemnogene abeparvovec, which is a gene therapy for the treatment of pediatric patients less than 2 years of age with spinal muscular atrophy, or SMA. SMA is an early onset, rapidly progressive, and universally fatal childhood disease, which results from biallelic mutation of the survival motor neuron, or SMN1 gene. The resulting deficiency of SMN protein results in motor neuron loss and progressive loss of motor function. The potential for gene therapy in neuroscience became apparent by the observation that the AAV9 serotype viral capsids have the ability to access the central nervous system when administered either systemically or through the cerebrospinal fluid and effectively transduce neurons and glial cells throughout the central nervous system. Following this observation, the first application was gene replacement therapy for SMA. Shown on the right are the data from the phase 1/2 trial and ultimately the pivotal trial for registration of this product, and is measured by the so-called CHOP-INTEND composite motor function score, shown on the y-axis. The dotted blue line below shows the natural history of the disease, with the loss of function leading to death within 2 years. And among the treated patients, the large majority showed a rapid and dramatic gain of function and transformational clinical benefit, some showing near normal gain of these functional milestones. Next slide, please. The next therapy is voretigene neparvovec, which is a treatment for patients with inherited retinal disease due to a biallelic mutation in the RPE65 gene. Now, RPE65 protein is a key component of the series of complex chemical reactions that make up the visual cycle. And when the gene is not functioning, the protein is not produced, leading to a progressive loss of vision and blindness. Voretigene neparvovec uses an adeno-associated virus vector of serotype 2, AAV2, to deliver a working copy of the RPE65 gene. This therapy is delivered by a subretinal injection at the back of the eye in a surgical procedure. And this introduction of the working gene allows RPE65 protein to be produced and the visual cycle to be restored, thereby improving vision. The results shown on this slide demonstrate a dramatic improvement in vision, which is sustained over 2 years and beyond. The clinical study of crossover design notably used a novel endpoint, a validated mobility test endpoint called the MLMT, the multi-luminance mobility test, which was basically a kind of maze that the patients walk through. And this is a measure of functional vision and a very good demonstration of their improvement. I also mention here that Spark Therapeutics is the sponsor of this program in the US. Novartis is doing the registrations outside of the US. Next slide. Another important example is tisagenlecleucel, an autologous CAR T cell therapy for patients with CD19 positive B-cell malignancies, like pediatric acute lymphoblastic leukemia and adult diffuse large B-cell lymphoma. CD19 is a transmembrane protein that in humans is encoded by the gene CD19. This therapy is a CD19-directed immunocellular cancer therapy. It involves reprogramming a patient's own T cells, which are one of the important white blood cells, with a transgene encoding a chimeric antigen receptor, or CAR, to identify and eliminate CD19 expressing malignant and normal cells. The figure in the center of the slide shows the rather complex manufacturing process of tisagenlecleucel, starting with leukapheresis of the patient. This is removal of the blood and separation of the white blood cells. Cryopreservation of the starting materials for manufacture. Shipment to a manufacturing facility. This is followed by the enrichment, activation, and transduction of the T cells with the lentiviral vector coding for the CAR, and the expansion and formulation of the product and quality release. This product can be considered a living drug, as once the cells are infused, CAR-positive, viable T cells expand in vivo in order to engage and eliminate CD19 expressing cancer cells. They may also exhibit immunological endurance to help support long-lasting remission. Tisagenlecleucel is approved in several countries and 2 indications, and we have a disease registry, CIBMT, including data from real-world clinical practice, which are depicted in the lower figure and document the important benefit this therapy offers for patients in a disease setting of high unmet medical need. Final slide. In summary, I hope in this presentation, I've been able to demonstrate some of the unique characteristics of advanced therapies, which address disease in a fundamentally different way than the therapies of the past. These therapies pose unique challenges in clinical study design, again, particularly with those very rare populations. The manufacturing and testing is highly complex and does require regulatory flexibility, especially with respect to change management and comparability. Successful development of these advanced therapies will rely on even closer collaboration and communication between sponsor and health authorities, and we look forward to working with you closely to bring these therapies to patients in need. Thank you very much. And from there, I will hand it over to Marae.
Thank you very much, Abril. So I will be walking you a little bit through the EU regulatory framework for advanced therapy. If we can go to the next slide. So the legislative efforts for ATMP started in the early 2000s. We have seen in Sue's presentation that we had a real evolution on the availability of these products, and this effort culminated with the ATMP regulation, but there were a couple of directives before the issuance of this ATMP regulation. It was important as it defined what ATMPs are and what they are not, that they belong to the biologics, and de facto they are granted centralized procedure. It also defined the Committee for Advanced Therapies, which is called the CAT, and it was a collective effort because the Commission involved and still involves through public consultation the broader public to comment on the regulation. Also, this is done on a regular basis to see if the regulation is still fit for purpose. If we go to the next slide. So as I already mentioned, the ATMP regulation defines what is important for marketing authorization, how you demonstrate quality, safety, efficacy on these products. And it defines additional features. But I would like to focus a little bit more on the specifics of ATMPs due to their complexity, their technical specificities. We needed to have really a tailored approach. The risk-based approach for pharmacovigilance, for example, traceability, as Auryn highlighted previously, as well as specific requirements for pharmacovigilance, long-term follow-up. These products have a curative potential, and you have to follow up patients for a very long period. On the other hand, good clinical practice and good manufacturing practice need really to be tailored for this type of products. If we go to the next slide. Yes. In addition, as highlighted by Auryn, we have seen in the 3 examples that these products often are meant to be used by a pediatric population for an orphan condition. If you know a little bit about the regulatory system in Europe, we have specific committees which are working on pediatric matters and orphan matters. The Pediatric Committee for Pediatrics, the Committee for Orphan Medicinal Product called the COMP for Orphans. If you take this all together, we have for ATMPs the involvement of 4 of the 7 committees the EMA is composed to. I'm afraid I lost the slides, but that's not really an issue. So I will continue to talk you through this slide, hoping that it will come back. So this committee is the involvement of the 4 committees is important and adds to the complexity of the products which already are rather complex. In addition to that, I wanted also to highlight that often these products are getting granted prime designation, which is the equivalent, the European equivalent of breakthrough therapies, and benefit of an accelerated review in theory. However, they allow to involve health technology assessment bodies. I hope, okay, Jenny might have a connection problem, so I hope this doesn't change too much. Okay, okay, then let's see. I hope you will still be able to see somewhat the slides, or if Livia can turn on the slides, that would be best. Or Tony, yes. However, so just to continue the talk, the ultimate decision is really on CHMP. So CHMP has the final word when it comes to the decision making and the recommendation for approval for those products. As you know, CHMP is the scientific body, but the decision, the ultimate decision for marketing authorization is done by the European Commission. So if we go to the next slide, so we are, yes. We have other specificities which can be explained, which are really due to the complex legislative framework in Europe. One specificity on the conduct of ATMP trials is the necessity to have a genetic modifying organism pre-approved in terms of impact on the environment. You have to file an environmental risk assessment prior being able to conduct a clinical trial in Europe with ATMPs. So I believe that's one of the specific aspects. If you go to the next slide, please. As Orin already highlighted, the manufacturing of these products is quite complex. And here again, due to the different legislation in each of the member states, we have differences in the GMP interpretation across member states. And this and makes actually the results of testing quite, or the quality testing more complicated, as you have to comply with different requirements across the EU. The supply challenges are also to be highlighted, but I will go to that in the next slide, please. So this slide, I won't go over it in detail. Orin has explained that in great detail. However, I would like to bring here to the attention that not all member states have the expertise or the infrastructure to be able to handle these types of products adequately. And due to the European framework, cross-border healthcare is possible and should be even facilitated for these types of products due to the lack of expertise and infrastructure in the different EU countries. So with this, I hand over to my colleague, Chin.
Yep, thank you, Mireille. So I want to spend the next few minutes to talk about the regulatory framework in the US. Not as complicated as the many nationalities in Europe, but complicated because cell and gene products are that way. But specifically, I want to talk in some detail about opportunities for sponsors and companies to engage with each other, as FDA is very interested in being a collaborative partner in cell and gene development. So I think we all agree that this is an exciting and new area of promising treatments for the patient community, for us in industry, for FDA and other governments as well. But because it is new, the cutting edge science is new, it's important that we have early and frequent communications with FDA as we develop these therapies. What we want to do is share the science behind our innovations and dialogue about how best to proceed in conducting the studies needed to demonstrate safety, efficacy, and quality to support the regulatory approval of these transformative therapies for patients. Next slide, please. So in the US, cell and gene therapies are regulated at the FDA at the Center for Biologic Research and Evaluation, the same center that oversees vaccines, including COVID-19 vaccines, blood and blood products, bioengineered tissues, stem cells, and what we're here talking about today, cell and gene therapies. Specifically, their experts sit in the Office of Tissue and Advanced Therapies, OTAT, where FDA's clinical, non-clinical, chemistry and manufacturing and controls reviewers sit as they provide scientific advice and review the data to support the application for regulatory approval. The US regulatory provisions are built on the same legal framework that we have in place for biologics under the Public Health Services Act. So in the US, cell and gene therapies are considered a biological product, but with specific cell and gene therapy guidances that are being issued by FDA to help companies navigate the cell and gene development programs. All other laws within the US also apply to cell and gene. As Mary said, we have mandatory studies in children. We have a rigorous regulatory framework for how we engage and the timelines used to engage with FDA, and also a very rigorous intellectual property framework for the protection of clinical data and the patents that are associated with these therapies. Those topics are complicated as well and evolving as many other topics within cell and gene are as we move forward, and the health authorities are learning as we are. Next slide, please. So FDA has established a host of opportunities for these and other transformative therapies to benefit from certain expedited programs that are at FDA. Some of these programs pertain only to cell and gene, others pertain overall to transformative therapies. So to facilitate development, there is the Breakthrough Therapy Program. Mireille mentioned that Europe has the PRIME program. There is the Regenerative Medicine Advanced Therapies Program and the Fast Track Therapies Program. All three programs afford companies additional opportunities to engage with FDA during development. And each of these programs have specific criteria by which companies must qualify. FDA has had these programs in place for many years on the fast track, just for a few years on breakthrough and regenerative. We find in industry that these are very helpful programs, especially in these areas of novel therapy, where again, we are learning together. So FDA also has expedited programs for marketing authorization. There is the very important accelerated approval program, where the basis of approval is a surrogate endpoint that is reasonably predictive of clinical benefit. So these programs that use the surrogate endpoints oftentimes are shorter in duration and allow a program to proceed to approval much quicker so that there is access to the therapy sooner. And then with that program, there are mandatory follow-up studies post-approval to confirm that that surrogate indeed afforded clinical benefit. And then lastly, there is the priority review program, which shortens FDA's review time to 8 months from the standard review time of 10 months. With these expedited programs in place, and a particular program can access one or several of these opportunities in order to bring a therapy to market sooner. Next slide, please. So in talking about opportunities to engage, FDA has also put in place a host of meetings to help address specific types of questions during development. So they range from what kind of therapy do I have? Is it a cell and gene therapy or some other? And this is not always clear, again, because we are all on this journey together, and there is not as much experience as with traditional therapies. Meetings with FDA, CBER, Advanced Technologies team in order to discuss new platforms, new methodologies, new technologies that might be used across multiple programs. The Interact meetings, which are early development meetings to discuss programs to establish safety and sufficient signals for efficacy to even proceed into our first in-human studies. And meetings for full development once we are in our human studies under the IND or investigational new drug applications. Device meetings when there is an associated device with the therapy. And then in full development for scientific advice and where we may have a stalled program and need to come into the FDA to receive special circumstance exemption or how to proceed forward. Next slide, please. So I come to my last slide. I mentioned that although cell and gene therapies are regulated under the biologics legislative framework, they have their own complexities, their own science, and they require their own set of guidances in order to be helpful to the industry. So FDA continues to issue cell and gene guidances as they are learning. Sue mentioned that there has been an explosion of activity within cell and gene over the past few years. And as we learn, FDA is learning, these guidances for general non-clinical and clinical studies and chemistry and manufacturing have been in place for a few years. But as FDA gains experience in specific disease areas, we're seeing guidances in the neurodegenerative space, as well as in the ophthalmology space, disease areas that Oren had touched on. So we look forward to continue collaboration with FDA as we bring new therapies forward. These transformative therapies are not easy to develop, but our attitude is that this is a collaborative effort with the patients, with the health authorities, and with the governments around the world. So with that, I hand it over to Livia to take us through the Latin American region.
So, thanks a lot, Ching. So moving forward here, I hope everybody is hearing me well. So moving forward here. So as Ching said, I'm gonna present this topic from the Latin America perspective. So I'm going to briefly address Argentina and Brazil, which are the countries in the region that have established regulatory frameworks in advanced therapies. I will start with Argentina. That was the first country to release an advanced therapy registration guideline in the region. That it's the provision 179 from 2018. So this provision identified these products as advanced therapy medicines. I think you may have seen, and I think Sue also mentioned that in the beginning, that it may varies a bit on the terms on how these therapies are called from country to country, region to region. So the provision 179 resembles a lot the EMA regulation mentioned by Mireille earlier. So the regulation 1394 from 2007. And similar to EU and US, these therapies are considered biological products in Argentina. They are assessed and authorized by the biologics area, and they are subject to the same regulatory principles of biologics. However, this guideline includes some additional provisions to address the complexity and the specificities of these therapies. Again, resembling a lot on what is the regulation in EU. So for example, there are some requirements related to traceability, to labeling. This guideline, another important point to mention is that this guideline also brings some flexibility by considering a risk-based approach to determine the extent of quality, non-clinical and clinical data to be presented. Because as has been shared by my colleagues previously, there's no one-size-fits-all. Let's say there are a lot of variety of products and platforms and all of that. So having this risk-based approach and this flexibility is something really interesting, really important. It is valid to say a bit in line with what Mireille and Chin said, that this provision in Argentina was a result of a public consultation. So there was a draft that was published by UNMAT, and it was gave to the sector, to the regulated sector, the opportunity to comment on that. Next slide, thanks. So in the same year, in 2018, UNMAT also released an updated GMP guideline addressing some ATM, so some advanced therapy specificities within the biologics annex. And a bit later, in December 2019, UNMAT took a very interesting approach by issuing a circular letter where they listed somehow considerable list of international guidelines, again, mainly from EU, on different topics. So for example, guidelines from EU related to production, to control, to surveillance. Some of them mentioned briefly by Mireille earlier, that should be considered as reference documents for advanced therapies in Argentina. And according to UNMAT, this list will be updated as needed. So moving to the next slide. Well, then I'm gonna move to Brazil. And then, next slide, please. In the case of Brazil, we have a bit different setting, let's say, compared to what has been shared so far. So in the registration guideline published by Anvisa, that it's the RDC 338 from 2020, Anvisa considered this product as a new category of medicinal products. So what means that they not fall into the biologics regulation in Brazil. And they are regulated, authorized by Anvisa blood tissue cells and organs management area, and not by the biologics area within Anvisa. Anyway, it is important to mention that the principles of existing medicines legislation, some of other principles apply to them in the same way that we see somehow in EU and US. So for example, to mention here, like GMP, pharmacovigilance, and this, similar to what we see with other medicines as well, this RDC also, for example, foresee as well the possibility to have discussions with the health authority prior to the submission, so what we call the pre-submission meetings. And, however, we have some exceptions. So, for example, different than what Chin and Mireille just shared that happened in EU and US. In the case of Brazil, the rare disease guideline and the priority review guideline that Anvisa has in place for the other medicines, they do not apply for the advanced therapies. So the RDC 338, that it's the advanced therapy regulation, as I mentioned before, it includes the priority review requirements that are applicable to these therapies, and also interestingly establish a conditional approval pathway, which means an approval conditioned with a later presentation of additional effectiveness data. That so far does not exist to other medicines. So this conditional approval, it's exclusively until now, at least in Brazil, for advanced therapies. Again, similar to other countries already discussed, this resolution was a result of a public consultation and of a considerable engagement between Anvisa, industry, academia, and other stakeholders in Brazil. Next slide, please. So a little bit earlier, so a couple of years earlier than the registration guideline was issued, so in 2018, Anvisa released two initial advanced therapy regulations, the RDC 314, that is related to good practice on human cells for the therapeutic use and clinical research. This resolution establishes minimum technical sanitary requirements related to the production cycle of cells and to the advanced therapies. And also the RDC 260 that determine the rules for conducting clinical trials with advanced therapies, which is has a structure pretty similar to the clinical regulation in place for small molecules and biologics. So both regulations, again, was also published as draft guidelines and had and the sector had the opportunity to comment on them. I would like to finalize this slide mentioning that, pointed out that a technical chamber in advanced therapy known as CAT was created in Brazil back in 2012. So it means that the advanced therapy somehow is a topic that is being considered and discussed by Anvisa for almost 10 years. And this chamber, together with the more recent created network of advanced therapies specialists that it's known as RENERA, has assisted Anvisa with the regulatory framework and as well with the evaluation of clinical trials and registration of new products in Brazil. So we are now looking forward to the end of our presentation today. And I would like to close providing some final message to the audience. So here, I would say this slide is a bit of a summary of what has been shared with all of you in around the last hour, let's say. So these therapies, as already said, but just to reinforce, they are very promising for changing the course of diseases and even potentially curing certain conditions. And on top of that, they are also promising because they work across multiple therapeutic areas, and their platforms are capable of different medicines. I think as shared a bit from Sue in the beginning of the presentation. On the other hand, I think as mentioned pretty well by Aureen and also with some insights from other colleagues, it presents some challenge for being an emerging and rapidly evolving field of high complexity. And as shared by Aureen, manufacturing and testing of advanced therapies is highly complex, and these therapies pose a unique challenge in the clinical study settings. So next slide. So here, just to close, I would like to share with you some foods for thoughts and with some recommendations. So as we have heard, advanced therapies is a reality, right? I think we some moments say that it's the future, but it's actually a reality. I think as Aureen shared and Sue presented as well, like we already have products registered, and it's definitely a lot coming up in the next few years. So it is important to establish a framework to bring these novel products to the market. and to consider leveraging parts or all regulatory and legal framework already in place for biologics, and tailor them through guidance, scientific advice as needed, and considering the specificities and complex that has already been mentioned. Also to consider the adoption of international regulations and guidelines. I think one example is, I think, the interesting approach that I, for example, mentioned about Argentina doing that. Be flexible to accommodate advances in science and growing experience in the field. This points together with the fact of that no one size fits all for these therapies, to have a risk-based approach while maintaining a robust set of key criteria to ensure patient safety and efficacy. It's extremely important to accommodate the evolution and the learnings that we are getting as we progress. And another topic, convergence, harmonization, and collaboration is crucial. So having a streamlined approach across agencies and geographies is encouraged and should be promoted, because I think Mireille brought some examples on having differences on how additional complexity this may bring. And adoption of reliance procedures should be explored here as well. And having a consistent open dialogue among all stakeholders is really encouraging and important. I think some of other colleagues has mentioned that before, and I would like to reinforce this as well. So I would like to mention that most of these recommendations that I just ran through are also in line with a position paper on unlocking the potential of advanced therapies for patients in Latin America that was issued last year by FIPharma, which is the Latin America Federation of the Pharmaceutical Industry. And the FIPharma position supports another somehow recent document that was issued by PAHO. It is the PANDRA Regulation of Advanced Therapy Medicinal Products, Concept Note and Recommendation, which is another valuable document with recommendations to the national regulatory authorities on this topic. So I would encourage you to check both of these documents. So I think now we look towards the end of our presentation. I would like to thank FIPharma for the opportunity to present about this exciting topic. It was I would say, a high-level overview for such an innovative and complex field. And now we would be happy to address the questions that the audience may have.
Muchas gracias a todos los expertos por traer tan relevante información a la mesa el día de hoy. Estimados asistentes, ingresaremos ahora a la sección de preguntas y respuestas. Recuerden, por favor, que pueden realizar sus preguntas y comentarios en idioma español y nosotros haremos la traducción. Como moderador, estaré a cargo de leer las respectivas preguntas en idioma inglés. Les recordamos que la interpretación en ambos idiomas se mantiene habilitada para nuestros facilitadores y los expositores darán respuestas según corresponda. En caso de que por cuestiones de tiempo no podamos cubrir todas las preguntas y dar respuesta a ellas, las mismas serán enviadas por correo electrónico, en donde se compartirá también la grabación de la sesión. A continuación voy a cambiar al idioma inglés para entonces dar inicio a la ronda de preguntas y respuestas. Okay, so we have several questions here for all of you. The first one is, what is the process that you follow in order to select a disease to further investigate as a potential advanced therapy? I think that maybe, Orin, can you help us with this one?
Sure, I can start, and maybe my colleagues may have some thoughts on it as well. So that's a very good and also a complicated question. Thinking about our own company, we have a very active early development part as well as a later development part. And for both of these, the disease strategy is given a great amount of thought, which disease areas we want to be in. And this also evolves year to year and over time, thinking about this strategically. And also looking carefully at whether an innovative treatment is available that holds a high degree of promise. In addition to in-house discovered products, we also look at potential in-licensing because a lot of these originate with small companies and in academia. So that's also very active for us, as I'm sure it is with other pharmaceutical companies. And when we think about getting into a new disease area that we haven't been in before, especially if it's a rare disease, we have to look very carefully about whether we're the right company, whether we can move quickly enough, whether we have the right expertise to do that. And that's an important decision too. So those are just a few of the factors. And of course, meeting unmet need of patients is at the top of the list. And again, determining whether we're the right company to carry those through. Maybe someone else has some additional thoughts on that.
Hi, Oren, this is Sue Stevenson. Just to add to that, no, absolutely right. I sit in the early drug discovery research area where when we think about starting a new drug discovery program, we look at all of the aspects that Oren highlighted. Medical need, could that particular indication be approached? What kind of modality would be needed? And when we eliminate that it can't be approached with a small molecule or a conventional biologic, there has to be a strategy for why an advanced therapy modality needs to be developed. For example, gene replacement. If there's no way to correct that gene or deliver or from a small molecule or protein replacement, then we seek to develop a gene therapy approach. In ex vivo cell therapy, a similar consideration. And so we only think about advanced therapy approaches when there's no other alternative. And then, of course, in settings of great unmet medical need when there are no treatment options available. So.
Great, thank you very much for both of you. We have here two questions that we can put together. The questions are, what has been the experience and also the opportunities that you have found or identified in Latin America for the registrations of these type of therapies besides Argentina and Brazil? I think that maybe Livia, could you help us on this one?
I would say I'm not sure, Oren, if you wanna, from a more practical perspective to start, and maybe if needed, I can complement.
Sure. So I happen to work on Veredogene, Naparvovec, the one for the inherited eye disease. So I'm most familiar with that program. And of course, going past those initial approvals in the US and Europe, we've been branching out over the world, and it's a limited number of countries, not the usual large number, because of the characteristics of this program needing specialized retinal centers, etc. So for Veredogene, Argentina and Brazil do represent the most current registrations, and we have approvals there, which are the end result of a lot of discussions with those health authorities and in those regions, trying to clarify and elucidate the regulatory pathway. So we are gradually expanding beyond that, looking at other countries in South America, Central America, and the Caribbean, and as well as in other parts of the world. And each time we go through, there are unique new questions and issues that come up. If there is less understanding, we try to work with the regulator and physicians in those countries to increase the understanding. So we're still early on in that process, but we do have filings planned and in progress in the region.
Great. Excellent. In line with the previous question, there is one that I think that we can cover right now is, there is any advance of advanced therapies regulations in Mexico based on that you mentioned before?
I can take this one. Yeah. No, we aren't, as far we are aware, there is no advanced therapies regulation in Mexico by now.
Excellent. Great. Okay, the next question is, what are the strategies that the industry intends to establish to achieve that these type of therapies cover more sector of the populations and are accessible to more patients due to the complexity of production and the cost that this sometimes implies? I don't know who wants to take it, to talk a little bit about it.
Well, maybe I could start. I think some of this may relate to manufacture and testing. For example, for Viredigene, we also have to take this as a cold temperature, minus 65 degree product. So we have to take a rather complex manufacturing and supply chain into account, one that a low temperature is one aspect, and also multiple sites as we move from drug substance, drug product, formulation, packaging, etc. There are certain product labeling challenges that we faced because we can't create as many different versions of the product as we might be able to, as we do for conventional therapy. So I think that is one aspect. As the manufacture and production becomes more routine, and also as regulators and countries gain more familiarity with these kinds of therapies, it may open the door to broader distribution and extending the reach, getting it to patients even farther across the globe. For products like Tisogen, like Lucel, that are autologous, and this involves removing tissue from the patient, that also has its set of unique challenges that may also create some limitations right now. But I think we will see this increase over time.
Great, excellent. Okay, moving to the next question. Here we have, given the complexity of the manufacturing, and beside this is patient by patient type of therapies, could you provide an estimated time frame or timelines from the production and the final administration once the patient is identified?
Yeah, I can take that. It depends, of course, of the stage you're within development. In the clinical studies, the turnaround time from leukapheresis or from initiation to manufacture to infusion back to the patient may take a little bit longer. The target time is really 22 days, 23 days. There are slight differences between the card developers, but in essence, of course, you need time to manufacture, you need time for the release testing, for the shipment. So 3 weeks is a high level ballpark, plus minus.
Great, excellent. The next question that we have here, it is for Livia. So regarding the Central American countries, even if some of them have biologics regulation, what do you consider is the first steps that the regulatory entities need to do to implement and guarantee the proper registration of this type of therapies?
Sorry, I was having issues to unmute myself here. So can you just repeat? So they are asking considering the biologics guidelines. So just to be sure that I understood it correctly.
Yeah, I mean, some of the countries in Central American region have already biologics regulations in place, but what is considered must be the first steps to adapting or to guarantee the proper registration of the advanced therapies in these countries.
Yeah, I think a bit on in line with what we mentioned, right, is to really see and leverage these guidelines, right, and see how they may be able to fit to accommodate not only the biologics and also the legal frameworks that are in place, and on how they can accommodate these therapies. And also, I think one important point that has been mentioned, I think in different moments during our presentation, is to be flexible, right? To really accommodate the specificities and the complexity. I think have an open dialogue. I think an open communication with the regulated sector, and like be willing to having us, and I think a bit relating with what Aureen I think shared, is like to really be open for us to construct a bit of this together, right? And have all this interaction, and for us to share learnings and all of that, because I think the experience that we have had so far was a lot based on that, on us engaging with each other and supporting this journey, I would say. So I think this would be the point for me. And for sure, I think as I mentioned, maybe also really considering how framework how eventually the authorities can rely and leverage frameworks that are already in place, and not build something from scratch as well, right? So I think, as I mentioned, a very interesting example of Argentina really just relying on what has been done in Europe, for example. So I think this would be from my side. Not sure if any of my colleagues want to complement on that as well, from their more practical, let's say, experiences.
Great. Okay, I think that we have, sorry, Aureen.
Oh, I was just going to add, I agree with Livia's comments, and I think in terms of, where do you start? I think gaining an understanding, a deeper understanding of these products and how they differ from the conventional therapies is an important baseline for regulators to understand how to regulate these products. So I think taking the time, and if the sponsors can do their part to help that process, then all the better.
Great, excellent. I mean, we have time for one more question. The question is, what kind of access considerations are taken into account at the moment to planning AT clinical trials model, or clinical trials model for advanced therapies, regarding price, tailored treatment according to Latin America patient profile, special prognostic, predictive factors, etc.?
Maybe I can start. You have a group of regulatory people, so we're not the market access experts, and I think we could probably have a whole separate webinar on that topic. But just like these therapies pose regulatory challenges and shifting paradigm, it's the same thing with market access, that these also pose new challenges for patient access. They're complicated, and we do have experts who are thinking about this from the earliest stages of development, and increasingly the regulatory and market access pieces are being looked at jointly. So I would only say it's a major consideration for these programs, and maybe we can look at that in a future discussion in more detail, because there's a lot to discover there.
Excellent, great. Well, we have finished the Q&A session. Now I'm going to change to Spanish to the closure. So just 1 second. Excelente. Bueno, habiéndose agotado el espacio para las preguntas y respuestas, no me queda más sino nuevamente agradecer a todos los expertos y ponentes por tan enriquecedora jornada y los conocimientos compartidos durante la mañana. Hemos llegado al tiempo programado para esta actividad, no sin antes recordarles que las preguntas que no han recibido respuesta por cuestiones de tiempo, igual serán contestadas y atendidas por correo electrónico. Cedo ahora la palabra a Diego Salas, coordinador regional de asuntos regulatorios para Fedefarma, quien dará las palabras de cierre para esta actividad. Muchas gracias a todos. Diego, adelante.
Muchas gracias, Douglas, y muchas gracias a todos los asistentes. En nombre y representación de la industria farmacéutica de investigación y desarrollo representada por Fedefarma, agradecemos a los jerarcas y funcionarios de las autoridades reguladoras que nos acompañaron, a los representantes de las industrias farmacéuticas nacionales e internacionales, a los gestores de registros sanitarios, asociaciones de pacientes, miembros de la academia, estudiantes y profesionales de la salud en general. Las terapias avanzadas constituyen un área científica en constante evolución, con un considerable nivel de complejidad técnica y reconocidos desafíos. Fedefarma, así como otros actores, vislumbramos 3 áreas de oportunidad que consideramos claves para fortalecer los sistemas regulatorios para estos medicamentos en la región. Construyendo sobre lo que indicaba Livia López respecto al documento de posición de la Federación Latinoamericana de la Industria Farmacéutica FIFARMA y que Fedefarma ha adoptado, y además en consonancia con las actividades de la Red PARF, a fin de facilitar el uso de terapias avanzadas aprobadas y minimizar sus riesgos para la población, Fedefarma considera y sugiere a los tomadores de decisión que establezcan marcos regulatorios consistentes con estándares internacionales, adopten procedimientos adecuados de reliance regulatorio e implementen programas de capacitación específicos para profesionales de la salud y pacientes en este tipo de tecnologías. Las terapias avanzadas tienen el potencial de transformar vidas y mejorar la eficiencia y sostenibilidad de los sistemas de atención médica. No obstante, requieren nuevos enfoques para la coordinación de la atención y la gestión de datos con estas nuevas tecnologías. A la fecha se encuentran en desarrollo clínico cerca de 800 programas para terapias avanzadas. Por lo tanto, la gestión de la incertidumbre inherente a este tipo de tratamientos y el diálogo temprano y de calidad entre los distintos actores del sistema de salud será fundamental para permitir que estas terapias, cuya evidencia marca un aporte de valor sin precedentes para los pacientes, puedan ser incorporadas a los sistemas de salud y lleguen a quienes las necesitan, siendo imprescindible la implementación de políticas claras y la colaboración proactiva de todo el sistema. En Fedefarma, creemos firmemente en la importancia del trabajo en conjunto con todos los actores del sector salud para construir un sistema de salud más sólido, moderno y sostenible, así como solventar las necesidades actuales y futuras de acceso a la innovación que son esenciales para mejorar y garantizar la salud y seguridad de los pacientes. Les invitamos a seguir a Fedefarma en nuestras redes sociales, Facebook, Twitter y LinkedIn como Fedefarma y en nuestro sitio web www.fedefarma.org para que puedan obtener más información de nuestros eventos y huellas en la región. Nuevamente, muchas gracias a todos por su participación, saludos cordiales y un feliz día.